so that we keep on developing. We have not reached the ceiling as physiologists in South Africa. The sky is the limit. You know, when I look at the audience and how integrated we are as physiologists, it's amazing how far this country will go with us. Let's take our position and take this country to greater heights in terms of the science that we are going to produce. Welcome and enjoy yourself. Thank you.
Thank you, ladies and gentlemen. Can I, before we proceed with the announcement, may I have the following keynote speakers with their presentation ready? I think the first one is Professor C. Smith. It's loaded. And also, is it Maugani? Is it Professor? Did the Professor Maugani? Oh, okay. Is it ready? Oh, we will offer them. Okay. Thank you, ladies and gentlemen. Those are the wise words from our PSSA president, Professor Muzi Masuku from Wits. In my program, we need to get the basic right. So I will allow the chairperson, the LOC chairperson, just to give us the house rule, and then we will take it from there. Okay, over to you, Lawrence. Yes.
Yeah, we're not all here yet, but I see many familiar faces, and Prof. Maseko summed it up well. And when I collected Prof. Maiberg yesterday, she said, it's not a physiology conference unless, and then you look for specific names, you look for faces and so on. Some people that we got used to through the years, because this is a family. And if you miss this opportunity to fit in, shame on you. The opportunity is here. You have ample time to sit down and talk. There's a whole room full of like-minded individuals who knows a lot about physiology, who make this their space. This is where they move. This is their life. Use them. There's Prof. Kathy's over there. I don't think there's many people taller than her. So just look for the shortest person and you will find her. A mountain of wisdom and experience in physiology. Many years ago, about 30 years ago, when I joined the society, she was already a recognized force in physiology. So there's really a proud history on that side, and we are so privileged to have you here, Prof. Maiberg. It's really awesome. Some of our other colleagues, Professor Kennedy from Wits, I don't know if he's here yet, I don't see him yet, but I think he's on his way. Those are the names that underlines and underscores our legacy as physiologists. And on Saturday, when I collected one of the students who's doing a PhD, he said to me, I wish that one day I can make a difference and also have a legacy. Do you even know what you're asking? Because it's such big shoes to fill, because it means commitment, it means time, not here and gone. It's a 15, 20, 30 years commitment to grow and make a difference. And if you're willing to do that, this space of physiology can be so rewarding. It's like in crazy. Those who studied at the Northwest University here in the early 90s would remember, it was still called the Potchefstroom University. In the physiology department, we had a professor who was already very old at that point in time. He was also in 1971, he was one of the first, I think he was the vice chair or vice president of a society, Professor Peter Pretorius. And he was known for always saying, physiology is a bottomless pit. You will never reach the end. There's always something more. It all depends on how far are you willing to go to find it. So, as our president said, he had humble beginnings and so on. It was difficult times, but many of us were all the ones standing here. We see many of our students sitting in front of us as professors. And that is the testimony that we have, that somewhere years ago we did something right. There's not much more where you can test how well you did. So for him, referring to me teaching him in those years, Professor Majani is also here and so on. Many of you came from the University of the North at that time. So it's indeed a privilege for us to be in this position to host you and welcome you to what I would call the most beautiful province, but I think Prof. Smith would differ from me because there's a mountain where she comes from that's seemingly quite nice. Got lost all around it. Everybody says, if you just watch the mountain, but you're okay. There's a mountain. If you see the mountain, the only thing I know about the mountain, I'm still in the Western Cape if I see the mountain. But the sea seems to be everywhere but where I want it to be. But nonetheless, this is a beautiful province. We've got good weather for you. It's summertime, temperatures in the 30s and so on. Lovely swimming pools if you want to use that. And if you want to network over there to swimming pools with something cold in the hand, this is your conference. You decide on how you will enjoy it. Just a couple of basic requests from our side. The venue arrangements are the following. This will be our main venue, Warrior, and then just on the other side you will see a smaller venue called the Day Lily. That will be of the parallel venue, a smaller one. So just keep in mind that tomorrow the Wyndham candidates, you will be here, and then the Johnny van der Walt candidates will move over to Day Lily. But we've planned the program in such a way that all the participants in the student competition start here tomorrow morning. We opened the morning so that the Johnny van der Walt candidates can sit here and see what it looks like when the big guns take each other on. Yes, we're a family, but sometimes, you know, we just take the gloves off and we just test the water a little bit, and at the end of the day, we still leave as friends. So, game on. Everybody's excited about it and nervous. I saw a couple of people running around being crazy nervous. It's not going to change, ever. You will always be nervous, so get over it. It's going to be there. If you come from a medical background, maybe a beta blocker can help. Half a beta blocker, I don't know, maybe. But don't lose your story, just lose the nerves. That would be good. Then, for the posters, we're going to put up a exhibit at the back on a PowerPoint roll that will roll over on all the posters and so on. So the idea is to have him up for 5 minutes at a time, and then we'll go over to the next one. We will put up the general posters there, but also the posters that entered into the Johnny van der Walt competition. So I would like to ask, I still have a couple of posters outstanding for the general posters. If I can have those by 9 o'clock this morning, just email it to me. And then we have a cutoff of 12 o'clock this morning for the Johnny van der Walt posters. But also, if you want to send us, and I would actually recommend it, those of you who are participating tomorrow in the Wyndham and the Johnny van der Walt competition, if you haven't done so yet, please send us your slideshows. Then we can start to upload them and have them ready here. Our time is really packed tomorrow. I think there's something like 23 Wyndham presentations and 20 Johnny van der Walt's. So it's going to be a busy day, and we don't want to, you know, run around looking for posters and circling to get it and so on. So just support us on that. Then you will see there's a, well, smallish pinboard. Dr. Linda called it her Connect and Collaborate board. Never heard of something like that. Maybe some of you have seen it elsewhere, but it seems to be working. Now with the Popular Act, it seems to be the in thing. You decide on what you want others to know and who you would like to collaborate with. So there's a bunch of small cards that you can put up there. If you want us to share your email address with others or contact particulars, you put it up there. If you want to collaborate with somebody specifically, you mentioned it there. After the conference, the LOC will sit together, we'll look at those things and then put them all together and circulate them to you, that you can maximize your experience here and really get what you want from it. And then the bathrooms, always easy to find. You would think, nay. It's always easy to find, because sometimes they hide the stuff and you don't know where it is, because it's behind that thing, after that thing, before that thing, and before you know, it's almost too late. But in this case, it's in the back of the venues. In this case, you move past the tables, there to the back, you'll find it there, and in Daylily, I think almost it's the doors where you enter, it's to the right of them. So, very easy to find. And then, yeah, that's about it for now. We will announce things as we go on. Is this?
The presenters for, especially this morning, but also this afternoon.
Oh, yeah.
Must urgently take their presentations.
Yeah, the lady at the doors, Dr. Carleen Linde, she will be here in the chapel where there's no wedding. It's just our, what do they call it? War room. It's where the stuff happens. She's available until 9 o'clock just to get all your presentations that we can beforehand put it on a USB. And then we're going to, if it's too big, we have some people in the audience who have slideshows that's so big you can't even get it on a plane. I don't know how they got it here, but they got it here. But if you can email it, email it. If it's too big, that you can't email it, just walk over and do it. Don't worry about Dr. Linda's email address. You have mine. You can send it to me. I will just then forward it to her. Right, can we then start, Dr. Hendricks? Right, so if you'll excuse me, I have my very capable Dr. Sekota, my Dr. Sekota, my deputy chairperson. He's in charge of the venue, so he will take you from here on. I'll then run the things behind the scenes. And then, yeah, Dr. Hendricks, I don't want to take over from you, so have fun. Thank you very much.
Good morning to you all. I guess we are ready to start, right? I just want to say once again, good morning to everyone, right? And thank you for all being here today, and I believe we will have a very exciting day ahead, right? Bear with me, this is also my first conference that I need to introduce a speaker. So thank you very much, but we are quite excited. And yes, for those of you who don't know me, I'm Jacobus Hendricks, and I'm from the University of Limpopo, and from this very exciting department of Physiology and Environmental Health that have organized this exciting conference. So let us start. If the talks are uploaded, you know, I'm very honored as well to announce our first keynote speaker, right? And this is Professor C. Smith, and he will tell us today about his expertise in the field of activism in the water, zebrafish as a model organism in disease modeling and drug discovery research. Let us welcome Professor C. Smith. Thank you.
Thanks. Perfect. Thanks. Good morning, everyone. Thank you to the organizers for inviting me. Professor Maseko gave such an inspirational welcoming just now that I'm not sure why I'm the keynote today. Very well done. And I'm most definitely joining you as a patriot and an activist for physiology today. So I'm not sure if many of you thought you would go to a human physiology conference and then see a zebrafish on a slide. I didn't think so either, up to a couple of years ago. So those of you who went to the Copnest conference when it was around Stellenbosch a couple of years ago may remember there was a zebrafish symposium there. And really, while I was interested in starting a zebrafish unit before that, we really only started 2 years after that. So I'm standing here today not as a zebrafish expert, but as someone really passionate about the versatility and the opportunity that this model holds. And I think we can all agree that funding is a never-ending issue in research. And I think zebrafish in particular can bridge the gap between trying to run research in a low resource setting without compromising on research quality. And so really my talk today, why I'm calling it activism in the water, I really want to encourage and hopefully entice you into also trying your hand at modeling disease in zebrafish. And so I've put up this first slide mostly for the supervisors because I don't want you guys to stop listening. Okay, but students, if your supervisor isn't here, and make them listen. Okay, so we started up 3 months before COVID struck, which was interesting in itself. But those slides that I've put up is really what we started with and what we had to work with for 2 years. A couple of tanks with some fish from the pet shop. The JoJo tanks that was left over after the drought. And basically an oven. Okay, so you don't need a lot of resources to set up a zebrafish lab. You do get recirculating systems. We've had ours for just over a year. It's not a vital component. It does save a little bit on daily maintenance, but you don't need that. And you don't have to go big. We have a little desktop version, as you can see on the slide. And even a small unit like that, I think we are used to seeing online the international units where there's a room full of water. You don't need that. You can have a few tanks on a bench and have enough eggs or larvae to supply many, many projects. And then I think in a publish or perish environment, I'm sure all of you will like the idea or the information that you can generate data relatively fast. And I think partly why this is because zebrafish is kind of tissue culture and a complete organism rolled into one. You have the benefit of a whole organism, but the transparency like you would have in cell culture. So you can really go molecular to whole body in one little go. So for those of you who are not familiar with zebrafish, that's what they look like. And why they have become the new thing in vivo research and disease modeling is partly because they're really convenient to work with. They have high fecundity, so they lay lots of eggs. They have high genetic homology to humans. Their genome has been mapped in its entirety already in 2013. And we know that there are homologs for 80% of disease-causing genes in humans. So it's a very versatile genetic model also with which to model disease. My group love microscopy, and you can see why we like zebrafish. On the top, there's a genetically modified zebrafish, which is fully transparent, and at the bottom, just normal larvae. They are transparent early in life. So they really lend themselves to visualizing physiology and biological processes as they happen. And so if you do some immunostaining or also genetic manipulation, you can create these wonderful images of events at molecular and cellular level. I'll come back to that slide in a minute. It's supposed to indicate that you can monitor and quantify behavioral responses. And then also, this is microscopy of larvae, or parts of larvae at least, with no staining. So you simply pop them under the microscope and you can see life as it happens and your intervention as it happens. So perhaps the best way to explain to you how we use zebrafish is to just tell you a little bit about our story and how we went about it. So just in brief, my group studies regulatory system, maladaptation in chronic inflammatory disease, with the 3 systems at the top, but now we're going for the Olympics with 5. And I'm going to tell you basically how we start. So we do disease modeling, and I think all of us want to normalize maladaptations in physiology. So we all do drug discovery. I think many people are interested in ethnomedicines. And if you look at the pipeline of disease modeling and drug discovery, zebrafish literally fit in and can contribute at every step of the way, okay, with the exception of clinical trials. So we started with the basics, and I think especially for those of you who work in ethnomedicines, even just the very basic testing that you can do in terms of toxicology or safety are really useful to do in zebrafish, and I'll explain to you why. The table-like image is basically showing you what a toxicology visual result will look like. So we have increasing age of eggs, and then they eventually hatch to become larvae, so time goes down, and then from left to right, increasing concentration of whatever treatment intervention you have. And you can, just by looking at these eggs every day, you can measure survival rate, hatching rate as a readout for developmental delays or developmental aberrations. And then on the right, you can look at morphology. And I think this is where zebrafish has the edge on cell culture, and we can do cell viabilities in cell culture, but you typically do that for one cell type at a time. Okay, so you would have to teach a student how to get the hang of culturing a particular cell type. Here you have the whole organism, and depending on the morphological abnormalities you see with increasing dose, you can pinpoint exactly which organ will be affected. Okay, so that immediately gives you a multi-cell type readout for toxicity and potential target organ. The next thing we did was to do activity tracking. I hope you can see on the slide, on the left-hand side, you can see individual larvae moving in a 96-well plate. Yeah, just not? Yeah, okay. And then you end up with a little track on the right, and you can, we have onboard software with the activity tracker that can give you quantitative results for behavioral responses to various treatments. And there are very many robust assays already published and validated that you can use. I've put one example up there, which is a model of seizure or excitotoxicity. Some people will say epilepsy. So you basically induce excitotoxicity by inhibiting GABA receptor signaling, and you can rescue it by adding a GABA receptor agonist. So you get this in the red line, that nice hyperactivity because of hyperlocomotion or seizure, and you can then test your treatment for effect to normalize that. There are many different ones such as this. One of the first papers we did in zebrafish, well, the first one was actually in the COVID period on ivermectin, and just zebrafish behavior. We could show that on the left-hand side we have the veterinary formulation of ivermectin, and on the right-hand side the human formulation. And without even me telling you what this response tested, I think you can clearly see with the one on the left, when you have any response flatlining, you have toxicity. So we could show immediately that the veterinary formulation is potentially more harmful. Then moving on, as you become confident, you become bold. And I have to say, I'm not a microinjection expert. She's sitting right over there. So if you want to do that, talk to Tracy. You can micro-inject even eggs or larvae. Eggs are typically injected with morpholinos or plasmids to do genetic modification in early 1-2 cell stage. But you can also inject larvae. That's a video of Tracy injecting larvae at the top. And you can inject them with about 1 nanoliter at a time. So if you have a very scarce extract or something that you don't have a lot of, you can inject a tiny little bit into a zebrafish and still get a full body response assessment. I'm not going to present to you much work that was done in adults. We took small bites, so we decided to start with larvae and then work our way up to adults. But we also do injections in adults. It's quite interesting to keep them alive outside of water, but we can do that, so we can talk to you about that. So I want to maybe explain to you how we use zebrafish as part of a bigger program, because I think that will make it more clear when and how they are useful to use. So one of the things we are interested in my group is the microbiome gut-brain communication in inflammatory disease. And in particular, the relationship between anxiety and IBS. I think the students can all nod. None of us know or do not know someone with anxiety or IBS or both. Okay, so I think when I started my independent research career, I looked at the students around me, and they were all anxious. I didn't cause it, right? But they were high anxiety. So I started my career looking at anxiety. So I've been going at this for a while. And so we started looking at the brain and anxiety and so on. So when I'm just going to take rooibos as an example because we've done rooibos assessments across many of our models. So I think we've all done the self-free assessments to assess, for example, antioxidant capacity of any antioxidant. So we've done that for rooibos. Then we also confirmed its antioxidant capacity in neuronal cells, following the general cell culture methodology. And we could see the moderate doses of rooibos that we used limited ROS production in these cells. And when we go too high, we see what we think is a pro-oxidant effect, and then we lose that benefit. Okay, so I think all of you have seen something like that. So when we got the zebrafish, we thought, okay, let's see what that does in this model. And you can see again, we saw exactly the same trajectory. So now we know we have physiologically relevant doses to test in our model. So I think that is where it's a little bit different from cell culture. And I really appreciate cell culture, so I'm not trying to shoot that down at all. But when we do cell culture, we typically take the highest dose that doesn't kill the cells, right? So in using a complete organism, we can really have the physiologically relevant dose. So to prevent making overestimations in cell culture that then don't translate to in vivo application. Then looking at potential neuroprotective or neurological effects of rooibos, we did a couple of different things. We used the PTZ assay, which is the seizure model that I've already explained to you, and we could show, again, some doses of rooibos having benefit, which is in keeping with the calming effect that is anecdotally reported for rooibos. And then at the higher dose, we also lose that benefit. So it's a quite nice, quick readout. You can do this within a week with no prior training. Get a quick readout of where your therapeutic dose is. And then we also did a different test, which is the light-dark transition test. It's often used to simulate anxiety-like behavior, but it's also quite useful in just measuring responsiveness to a stimulus. So it will also reflect cognitive limitation, for example. Okay, so we used that. We induced an anxiety-like behavior, and we could again see that rooibos has a therapeutic effect in the same dose range. So stroke doesn't have much directly to do with anxiety and gut health, but I'm putting this in just kind of to be realistic about this model. You have to remember that it's a developing organism. So when you are interested in assessing a particular organ or system, you have to make sure that it's developed by the time you want to look at it. Okay, so the stroke model quite nicely illustrates that. So we induce stroke, and this is kind of a easy or a common way of inducing disease in zebrafish, if you don't want to go the genetic route, is by with pharmaceuticals. In this instance, we induced stroke using ponatinib. Ponatinib is actually a leukemia drug that was withdrawn from the market because it showed increased risk for stroke as a side effect. So now it's been adapted for use in zebrafish, and it quite nicely, oh, that sounds wrong, saying nicely with stroke, but successfully induces stroke. And you can see we induce it on day 1, which is still an egg stage. Okay, we induce it on day 1, and by day 2, they have coagulation factors, and they stroke. And you can see this is a hemoglobin stain. You can confirm that you have clotting in the forebrain of the larvae. Then, because we were working on rooibos, we were interested in an antioxidant testing in larvae. We've tried it on day 2, day 3, day 4 post-fertilization, and really you only get robust data by around day 4. So that's just something to keep in mind. But when you do it at the right time, you can again see how there's a response to the stroke, and there's a normalizing effect of rooibos. And then activity is also an interesting one, because you can only have a larvae properly swim when it has an inflated swim bladder, which gives it its buoyancy so it can stay upright. So we only that happens on day 4, so we don't do behavioral analyses before day 4. Okay, so you just have to know the timing of the different things you want to look at. Here again, you can see that when they stroke, there's an immediate reduction in activity. So there's that limitation that is normalized or prevented in the presence of rooibos. Whoops. Sorry. Then, and I think this is the beauty of the larval models. So we do a lot of our work just in larvae. Often you don't have to even section. You can just take the whole larvae, stain it, and pop it under a microscope. Okay, so this is what we've done here. And I wonder, is it possible to put the front light off maybe? I'm not sure if it's quite clear. Yeah, I have a few fluorescent images, but it's fine. So here we looked at 2 things in particular. NeuroD1 is a precursor differentiation marker in the brain, so it's very important for neuronal development. So it's expressed early, and you can look for that already at 2 days post-fertilization. So very early in your protocol, you would be able to see a response in that marker. And then tyrosine hydroxylase is kind of the golden standard for assessing dopaminergic neuronal development. And you can see that later on. But for both of those, we saw in response to oxidative neuronal damage, there was an increased pro-synthesis or pro-repair response in the stroke fish, which was not necessary in the presence of rooibos. So quite sensitive, easy to execute markers. We also, looking at brain and gut, we were also interested in responses other than just antioxidant function. So we were in particular also interested in barrier integrity, thinking of the gut-brain and the blood-brain barriers. And so we looked at cell junction proteins. And much like you would for cytokines, we just did diffuse staining in the brain area. And again here we could sensibly, the model is sensitively reflecting effects of stroke and your treatment. Then I'm going to move on to gut health. And we started looking at gut health in the context of IBS. And we were specifically interested, I was working with microbiologists at the time, and we were specifically interested in the role of the microbiome. I think many of us will know, working with ethnomedicines, there's this continuous criticism about ethnomedicines being poorly absorbed in the gut. And people are saying, if you can't detect it in the blood, how do you know it actually has benefit? So our hypothesis was that maybe it doesn't have to be absorbed. Maybe it changes the microbiome signaling. And maybe that is how you modify physiological processes from the gut up. Okay, so we were interested in looking at that. So the first thing we did with our microbiology partners is to set up some cultures of commensal bacteria and also some pathogens, and we exposed them to different formulations or preparations of rooibos. So we used an ethanolic extract donated by the guy who paid for the work, and then we also took aqueous extracts of both unfermented and fermented rooibos. So the pie charts that I'm showing you, the size of the pie chart is basically giving you a relative readout of the volume of secretome that we measured. And then obviously the little pies are different components within that secretome. And you can see how, depending on how you prepare the rooibos, it either has not so much of an effect or a huge effect on the proteins and peptides that these bacteria were secreting. And we were particularly excited to see agmatine and its metabolites. They're called trace amines collectively. Agmatine in particular has been linked to protection of barrier integrity in the context of the blood-brain barrier. So we were quite excited to see that rooibos could potentially do that for your gut. So the next move was to try this out in cell culture. The images on the left is showing tight junction proteins. So we know if the cells lose their connections between each other, you will have loss of membrane integrity or leaky membrane. And you can see the control. And then with the agmatine, we could see that it changed the expression levels of these tight junction proteins, but unfortunately also their expression patterns. And we could see large areas, I don't have a pointer, I don't think, but you can see like on the left upper corner, there's total disruption of cell borders, okay, in terms of tight junctions. And that is actually an undesired outcome, which didn't quite fit with the literature. So we were a little bit worried about that. We were delighted to see that if you added rooibos, it prevented that. So rooibos is still great. But we were a little bit worried about this agmatine that showed a negative effect. But then looking at the agmatine metabolic pathway on the right-hand side, we saw that some of its downstream peptides or metabolites actually have antioxidant capacity. And so we thought maybe what we need is for agmatine, when it's administered or when it's secreted by the microbiome, maybe it has to be metabolized before it has its benefit. Maybe it's not agmatine itself. And of course that you can't see in cell culture because all those metabolic enzymes are not available in cell culture. Okay, so we thought, let's try this in the zebrafish to have an in vivo model where you have clearance and metabolism intact. So one of my PhD students, Lisha, I think her picture was somewhere on the slides. I didn't mention her earlier, but she did a lot of the IBS work. She came up with this assay where you stain the larvae, whole live larvae, with neutral red. And that will stain basically the functional epithelium in the mid-intestine. And you can see it with a normal light microscope as the red portion on that slide. And you can quantify the length of the intestine, the intensity of staining. You can use the stained area to look at the size of the lumen, all kinds of other parameters, much like you would analyze an H&E section. So we just did, by looking at length of stained or functional mid-intestine, we could already see, even in the absence of a stressor, agmatine increased the length of functional gut. And then we also have a TNBS model, which is a simulated intestinal inflammation. And also in that model, it had a beneficial effect. So I think that is where zebrafish can improve the translation of these early preclinical testing to more complex in vivo models. So then we used the same TNBS model, looking at the effect of rooibos. And we looked at the length of mid-intestine again. The blue bar is prednisone. Prednisone is used as the standard treatment for, as a standard anti-inflammatory rescue in zebrafish models. And you can see the fermented not so much, but the unfermented rooibos treatments, both the ethanolic and the water extract, both had a normalizing effect there. And when we looking at the bottom graphs, we also saw benefit in terms of ROS production and prostaglandin E2 production as a proxy for inflammation. And then I think what you do and what you analyze in zebrafish depends very much on the expertise that's already within your group. If you're strong on genetics, you can do genetic assessments. We are strong on microscopy, so that's what we did. So the picture on the left is a whole larvae embedded in resin and then sectioned. And then we can come up with very much the same type of image as you would see in a cellular monolayer monocell one. I just lost my English there. A cellular monoculture. Okay. So the images on the right is basically a section through the gut, and it's basically in the form of a C, and you're looking into it at the epithelium on the inside of the lumen, and you can see very much the same as the previous pictures I've shown you of the tight junction proteins. So we've done that also in the zebrafish to determine cell junction correction with rooibos. So you can see how, depending on the technology you have and the instrumentation you have, you can get to the same answer with many different analyses. And then for me, the fun part is to link those molecular and cellular processes and the changes that we've affected, to link that to functional outcome. Okay, so in the live larvae, one of the things we can do is to look at, if you look at the area where the bracket is, you'll see a peristaltic movement. So you can measure the frequency of those, the size of the contraction, and quantify that as a response in terms of gut motility or spasm. You can look at pain. We have another nifty experiment on gut motility, where we simulate either increased or decreased gut motility, so relative simulations of constipation and diarrhea. And even in these models, so what you basically do is you feed the larvae fluorescent food and you measure the rate at which it's excreted. Okay, and you can do that in a plate reader. So using that, we could show that rooibos had benefits to normalize both diarrhea and constipation-like outcome. So now that we know hopefully a little bit about the brain and about the gut, we thought to pick a disease context so that we can start with interventions. And I'm not sure about you, but we have been reading a lot about autism. I think we all know someone who is on the spectrum. And they have 100% of people on the spectrum has anxiety to some degree, and more than 90% of them also have IBS-like symptomology. So we thought that we would do this. And so we are profiling microbial secretome in humans to align our disease model and our interventions with. And then we have been working on a genetic model for autism. And I'll just briefly explain to you how we do this, because you don't have to micro-inject and physically change the DNA. You can sometimes just also pharmaceutically induce a genetic model. And the autism model is one of those. So if you just look at these larval heads at the top, you can quite easily see from left to right three differences that's quite striking. The first one is the size of the snout or the mouth that changes with increasing concentration of treatment. So we use valproic acid as an inducer. Valproic acid is an anti-epileptic, which comes with the risk of ASD in offspring. Okay, so we know it has a genetic modification as a risk. When you treat larvae, you consistently induce autism spectrum disorder in them. Depending on the dose you use, you go kind of further across the spectrum. And we've picked a dose where you get a significant readout, but you don't end up with a completely dysfunctional fish. And I think this is where it's important to be able to do some microscopy, because obviously the ones on the right, where they have their snout too much deformed, they won't be able to feed properly. So if you wanted to raise them into adulthood like we're doing now, you have to have at least properly feeding fish. So that was the one difference. And then if you look at the distance between the eyes, the intraocular distance is a good indicator of initial hyperplasia in the brain that we see because of dysfunctional neuronal growth. And you can measure intraocular distance as a confirmation that you have induced autism. And then if you look at the behind the eyes, so above the eyes, those two pockets on the left and the right, those are the gills where gas exchange happen. And you can also see, if you go to the right, the morphology looks a little bit different in the gills. And that the oxidative dysregulation, and we do see in autism that there's significant mitochondrial dysfunction. Okay. Then we are currently doing some high-resolution characterization of this mitochondrial dysfunction. I've just put up one example of a neuronal marker that we've assessed, neuroligin 3, which is responsible for synaptic organization, and that is also aberrant in this model, and we could quantitatively show that with fluorescence. I think this is the last slide. Then in terms of anxiety, to just confirm, no, 2 more slides. We confirmed anxiety in the larvae using 2 different models. The one looks at light preference or dark preference. Anxious fish will spend more time in the dark. And also thigmotaxis is a very particular behavior where they kind of hang in the meniscus of the water. And if they are not anxious, they will explore into the middle of the well. So just looking at where in the well they are also confirm anxiety in the zebrafish. And then lastly, we repeated the neutral red gut health, and we also showed gut complications. So we're ready for intervention now. So thank you for listening. I hope I have managed to make you a little bit excited about zebrafish. You stuck with me and my group until fairly late on Wednesday, so please look for us, come and talk to us. We love collaboration. Thanks.
What a wonderful talk. The floor is now open for questions. We will allow 5 to 10 minutes of discussions. Is that okay, Prof? Can we have a mic, perhaps, if there's on the floor? Oh, thank you, Reis. Right, maybe I will start, Prof. I will ask a general question, because I have done all my studies on rats and mice. So perhaps maybe I will ask you, what is the advantages and perhaps also the limitations of using zebrafish, above rats or mice? Thank you.
Okay. I think the speed at which you can generate data is a big plus. It's definitely more cost-effective than keeping rodents. You can keep, your whole study in a little tank, whereas with rats you have several cages. So just in terms of maintenance cost and breeding cost and husbandry, you save a lot. I think the zebrafish model is a very versatile one. If you are used to doing cell culture work, you will love zebrafish work. You take very much the same approach as cell culture, doing kind of pooled samples in triplicate, for example, for quantitative analyses. And microscopy is much the same ends as you would use in rodents. I think some limitations are obviously if you work in larvae, they're developing, so you have to know when your system will mature. I can give you lots of literature on that. That's not a limitation, it's just a consideration. Yeah. And then obviously they're not mammals. Okay, we can't simulate birth in a fish. They lay eggs. But you can look at fetal development easier than you can in a mammal because their larvae or their fetus develop outside of the mother. So that's a benefit. They don't have lungs, but their swim bladders are very similar in terms of lungs. They have the same epithelial, they even have surfactant. So there's often a homologue for the organ system that you are interested in. So, yeah, I mean, we've been going at this for a while. So if it sounds foreign to you, just come speak to me, I'll convert you. Yes.
Thank you, Prof. Smith. That's very interesting, and I think very commendable what you have achieved, because we've been speaking to some of our collaborators who work on zebrafish, and we know how difficult it can be. So my question is more kind of after you've done with the research and you try to publish your paper, like we do a lot of animal studies, and we're always faced with this question of what is the clinical translation? So how do you answer this reviewer comment? What is clinical translation when you try to publish zebrafish research?
So I think Tracy has a talk a little bit later to demonstrate the homology between proteins in zebrafish and humans. We often use anti-human reagents, which you can't even do in rodents. So there's really high protein homology between zebrafish and humans. But just like any other preclinical model, you have to remember that it's a model of disease. You still have to continue all the way down the pipeline into human studies, but you can probe mechanisms with the benefit of a full body metabolism in zebrafish, which I think is less achievable in more complex animals. Just maybe on publication, we have not struggled to publish anything in zebrafish. We've been going for 4 years now, and in the starting from scratch with all of lockdown included in that 4 years. And in the last 2 years alone, we were able to publish 8 papers on zebrafish-based studies. So if you are kind of a little bit hesitant to start out this new endeavor, it really pays off fast. You don't spend years and years to become an expert before you get a paper out.
Thank you, Prof, for a nice talk. My question has been, one of my questions have been partly covered by Suraj. But I'm more interested in terms of how you identify different brain regions and perhaps harvest them so that you can do some biochemical analysis, as well as in terms of the zebrafish chart, how is it comparable to humans? Thank you.
So, sorry, I'm a little bit hard of hearing. You said you're interested in knowing how I identify what?
How would you go about identifying different brain regions in a zebrafish, and also in terms of dissecting them out to perform biochemical analysis?
Okay. So in the larvae are typically 2 millimeters long. Okay, so you don't really dissect out a larvae. We have dissected like the head off because the rest of the body is almost all muscle and cartilage at the early larval stage. So if you want to look for brain proteins which might get lost in the rest of the body, you would cut the head off, for example, and use that. Other than that, we would normally section the larvae, but if you use adult zebrafish, you can dissect them like anything else. The organs are quite easy to recognize. Even the parts of the brain in the zebrafish larvae is particularly interesting because it's not roundish like ours, it's kind of stretched out. And it has a very specific anatomy, so it's actually easier to dissect out parts of the zebrafish brain than parts of the human brain or rodent brain. They're very good atlases for that as well.
Thank you, Prof, for a very interesting talk. I'm also interested in disease modeling, looking at the effects of ethnomedicines in combination or monotherapy with different drugs. I'm just wondering, okay, so a couple of questions really, but I'll just try and limit it to 2. When setting up the zebrafish, I guess the lab itself, is this a small lab space that you require, or do you need to then collaborate with your animal unit to do the breeding? Like, how specialized or how complicated is it to set up yourself without the experience? And then maybe also just as an extension of that as well, in terms of getting the zebrafish or purchasing them from the fisheries, I think you said, what is the I think the ethics process around that? Is it something complicated? Is it a matter of consent? How does that work? And then I'll just leave my third question maybe for you, just to first answer the first 2. Thank you.
Okay. In terms of husbandry, our zebrafish unit is the size of an office. It's tiny. You need 1 bench and an incubator. So you don't need a lot of space. You don't need to be in an animal unit. I think there are very specific guidelines on doing infection risk, for example, but it's not excessive. If you send me a student, I can teach them how to do that in a week. You don't have to have special skills. You don't have to have special training. My group and I have done the husbandry course, which is the University of Alabama at Northumbria. They do a very good online course over 16 weeks, but in your own time. So we've done that, and other than that, we've helped ourselves. It's really not complicated to start up. It's not the same as looking after rodents and keeping strains going and things like that. Breeding, I think if your water quality is the major thing, you have to understand the water quality. If they are in good water, they will breed on anything. So, yeah, that's not a problem. In terms of ethics, the new SANS guidelines, previously, up until day 5, larvae were seen as not sentient animals, and they weren't protected by SANS, and you didn't need ethical clearance of protocols. The new SANS now protects even eggs, so you would need ethics approval for any zebrafish work. But typically we, and I will even assist if you have ethics committees at your universities to give them some guidelines in how to evaluate applications. You take kind of, zebrafish are a little bit between tissue culture and rodents, particularly when you work in larval models. You can't count single larvae. Okay, some of them just die because they were born abnormal. So if you count single fish, that becomes quite hairy. Adults, you would do like a rodent model, but for the larvae, you would kind of assess it like you would a cell culture protocol.
All right, thank you so much. And then the second question is with regards to disease modeling. So we work on cancer and then neurodegeneration as well. So I'm just thinking, you mentioned that you use larval samples, larval zebrafish. So if you want to, for example, look at an aging model, is it a matter of over time, how long would you grow the zebrafish for? And if you wanted to look at, for example, a tumor microenvironment, does it simulate that? Like, how would you actually induce that?
I think cancer is induced much the same way you would be in rodents. You can either do it by genetic manipulation if your particular cancer of interest has a significant genetic known cause, or you could even implant cells. So much the same way as you would do it in rodents. And then you asked about kind of aging. Right, so I think that is the one benefit I forgot to tell you. So zebrafish are one of the big benefits in terms of disease modeling and medicine's development is that you can follow zebrafish over the span of a whole, or over the whole lifespan. So typically zebrafish in nature, where they have many predators, they only live for about a year. In our lab, we cull them at 18 months because then they start to get proper age-related abnormalities like scoliosis, and they struggle with fluid balance and things like that. So you can quite easily, over the span of, I would say, a year to a year and a half, follow. I think by the time they get to 18 months, they are geriatric. So that's basically, it can fit into a PhD.
Thank you.
Please give a round of applause for Prof. Thank you very much. Our next speaker is Maugani VF from Northwest, Association of Angiosthetic Converting Enzyme 2 with the oxidative stress-related marker in young adult, the African PREDICT study. Please give a round of applause for. We managed to get a pointer. Prof, sorry, we just managed to get a pointer now. Just managed to get a pointer now. So next time.
Is it this one?
Yes.
This one, pointer, and the left and right, yeah.
Oh, I think I'm going to be fine without it. Yes. Good morning, everyone. My name is Dr. Felicia Morgana from the Northwest University. The title of my abstract presentation this morning is Associations of Angiotensin Converting Enzyme 2 with oxidative stress-related markers in young adults as part of the African PREDICT study. This is a brief outline of my presentation. Cardiovascular disease is the leading cause of death globally, and studies have reported a higher cardiovascular disease risk in men than in women. One of the contributing factors to the development of cardiovascular disease is oxidative stress, which is reported to be more pronounced in men than in women. Oxidative stress is defined as the overproduction of reactive oxygen species and a reduced antioxidant defense capacity, which causes damage to macromolecules. This figure illustrates the redox signaling, which consists of oxidative stress markers shown in red and antioxidant markers shown in green. Superoxide is converted to hydrogen peroxide by superoxide dismutase, and the end product, this end product is then converted to water and oxygen by the enzyme catalase and by the glutathione cycle. MDA, which is malondialdehyde, results from lipid peroxidation from polyunsaturated fatty acids. Oxidative stress is also linked to the renin-angiotensin system, which consists of the classical and the alternative pathway. Angiotensin converting enzyme 2 is a member of the counter-regulatory axis of the RAS. ACE2 limits the adverse effects of angiotensin 2, such as increased oxidative stress or overproduction of ROS and inflammation. It also degrades angiotensin 2 to angiotensin 1 to 7, which then reduces oxidative stress and increased nitric oxide bioavailability. ACE2 exists as a membrane bound and in the form of soluble, which can be measured in plasma. And a few studies have focused on plasma ACE2 levels in healthy individuals, where in healthy individuals, ACE2 levels range from undetectable to very low. Plasma ACE2 is emerging as a biomarker of cardiovascular disease risk, and circulating ACE2 is increased in hypertensive patients versus healthy controls. And studies have reported associations of increased ACE2 with adverse outcomes such as heart failure and myocardium fibrosis. Studies have also reported an increased plasma ACE2 levels in men compared to women. However, it is unknown whether oxidative stress-related markers associate with ACE2 activity and whether sex plays a role. The aim of the study is to investigate the associations between ACE2 activity and oxidative stress-related markers in young, apparently healthy men and women. Data was collected from participants of the African PREDICT study who were aged between 20 to 30 years of age. Aged between 20 to 30 years. We excluded participants with missing ACE2 data, and we divided them according to men and women. Oxidative stress markers and antioxidant capacity markers were measured in serum and plasma. ACE2 activity was measured in plasma using a validated assay. The results of the study shows that men had higher ACE2 activity compared to women, and women had higher ROS and MDA but also higher total glutathione. Whereas men had higher GGT and GPX, as well as total antioxidant status. After multiple adjustments for covariates, we have found that women had a positive, or ACE2 associated positively with ROS, total antioxidant status, and total glutathione in women only. Whereas in men, ACE2 associated positively with GGT and GR, and also in women, ACE2 associated negatively with SOD. The main findings of the studies are ACE2 associated with oxidative stress and antioxidant defense capacity in young adults, with unique associations found in women only. The association of ACE2 with ROS may be that ACE2 is increased in response to oxidative stress, and the positive association with total antioxidant status and total glutathione may mean that higher antioxidant capacity increases ACE2, which may also increase the nitric oxide bioavailability and may decrease ROS, which is a protective effect against oxidative damage. However, we also found a negative association between ACE2 and SOD. And SOD is also known as the first line of defense against the effects of increased ROS, and upregulation may be needed to counteract the effects of ROS or increase the dismutation of superoxide to aid in the prevention of oxidative damage. However, in addition to the upregulation being insufficient to prevent increased ROS, it may also be insufficient to preserve nitric oxide bioavailability, but not to the point that ACE2 is reduced. And over time, this may lead to the enhanced oxidative stress, because women in this study were found to have higher CRP levels, which is a marker of inflammation. So, and this may predispose this woman to increase future cardiovascular disease risk. In conclusion, ACE2 activity associated with ROS, total antioxidant status, and total glutathione, with a negative association with SOD, increased oxidative stress and decreased antioxidant capacity may increase ACE2 activity. Increased antioxidant capacity may increase ACE2 activity as a protective effect against oxidative damage. ACE2 activity may be useful in understanding sex differences in the pathogenesis of cardiovascular disease, which may improve cardiovascular risk prediction. The authors would like to acknowledge the participants who participated in the study, and the research team of the Northwest University Heart Department, along with the students and also the funders. This is a list of the references. Thank you so much for your time.
Thank you, ma'am. Thank you, ma'am. Open. May I have water? Can I have questions? Let's. Please, gentle one, the nice one. Katan. Oh, you will speak loud.
So the study few years. Have you done any relationship studies with the outcome and actually looked at the patients that got cardiovascular disease in this time to see any relationship with loss or with
No, in the African Predict, it's only the younger populations. We have never looked at people with. The African Predict is normally a study that is looking at younger or young adults. So we have never looked at other participants with cardiovascular outcomes.
and there's no one and you don't follow up these participants to see if
The follow-up is ongoing. We can look at that because this is based on preliminary results. So we can look at the follow-up data of the African Predict to see what happens.
One of your adjustments, when I looked at you, when you adjusted for the confounders, I saw waist circumference. Did you include BMI as well? Because men and women have a higher BMI compared to men, and this might have led to your findings that women are more predisposed to cardiovascular disease than men.
No, we did not look at BMI, but we can consider that because, like I said, this is based on preliminary results, so we can go and look into that as well, adjusting for BMI and replacing it.
Okay, thank you for your talk. A quick one. On your sex differences on ACE2 activity and oxidative stress markers, do you think lifestyle factors or environmental factors could play a role in the results that you've explained? If so, how?
Please repeat your questions.
Lifestyle factors or environmental factors, how do they affect your results?
Yeah, in this case, I think smoking is one of the lifestyle factors that is all. Thank you. That is known to be involved in oxidative stress, so we did adjust for that as well. We did look at it.
Thank you, ma'am. And please give a round of applause. Okay. Okay, thanks. The purpose of conference is to engage in this debate, but I'll encourage, let's engage even after the debates, after this presentation with the relevant presenters. I would love the following people, please approach the registration team. Gumete, Majida, Skosana, Chikupela for your slides. We need those things. Martin, please come and help here. The next presenter will be Prof. Nell. Oh. It's okay.
Right. Thank you very much for having me and for this prime spot to give my talk. I have to admit that I haven't been to a PSSA conference in quite a while, but I tend to send my students, and you've always been very kind in giving them prizes, so I thought maybe it's time that I present something myself. So I'm going to try to go through quite a lot of slides, so if I speak fast, my apologies. We've been working on a project for the last number of years, establishing a new model, a new in vitro model to really recapitulate cancer. And the goal here is really to start studying cancer in the Black African population, because there's a real paucity in information and knowledge about cancer in this group. Oh yeah, wait, it's like this. Okay, so why do we do that? So I think it's important to always paint this picture, because it's actually a pretty dire picture, if I'm honest. So these are some statistics for breast cancer, and I've basically juxtaposed African versus European populations here. And what you'll see is that there's quite a few differences, some of which are biological and some of which are going to be socioeconomic and environmental. So first of all, it's really important to notice that the incidence of breast cancer per 100,000 is quite different between the black population and European, white European population, in that black African women tend to present less with breast cancer. Now that can be partly due to just not going to a doctor and presenting, but also it can be biological in that there's just a lower prevalence of breast cancer. What's also really important, and this is the most striking for me, is that the presentation, the age at presentation is also vastly different. And so for black African women, it's mostly premenopausal, so an average age of 49, whereas for white European women, it's mostly a postmenopausal disease with an average age of 67. And that can really only be attributed to biological differences, because there's not really any environmental cues that might cause that. Then lastly also, you'll see on the right-hand side, can you see it? That the stage at which the cancer is when presenting is also vastly different, in that most African women will present with stage 3 or stage 4, which basically means that it's grown, invaded, and attached to the chest wall, and also very often metastasized, whereas in a white European woman, it's mostly stage 1 and stage 2. And a lot of that has to do with the healthcare regimes that the different groups populate. And then lastly, and most importantly also, the 5-year survival rate is really different, whereas for a black African woman, it's about 79, the percentage is 79 5-year survival, whereas 90 or above for white European women, which highlights the fact that we really need to pay attention to this. And so what we wanted to do is actually be able to pull out the biological differences and separate them from all of the socioeconomic and cultural difference that may cause these disparities in outcome, and build a model that we can really use to investigate the biology. So what we did, and I was just looking at the latest edition of the Cell Journal, and there's like 3, review articles and opinion pieces on in vitro models. It's a big field at the moment. Is we set up cancer organoids as avatars of patients. So these are new ways of growing cancer cells in vitro that better represents the tumor. And the most important part of that is that we can retain the heterogeneity that's inherent within the tumor. The other parts that are important is these are expandable and storable, so they can be used for multiple studies, and we can do a lot of different types of studies. So we can use them for different omics, we can use them for drug screens, etc. So in this project, we wanted to study the pharmacogenomics of chemotherapies commonly used within the public health system for black African patients, to see the differences in sensitivity to the chemotherapies as a way to start to stratify them and use these PDOs as a functional precision medicine tool. And so we established a small panel of these PDOs, assess a chemotherapy response, but also use them to start to characterize them in terms of their biology, specifically their metastatic potential. So what that looks like in theory is we have a patient with a tumor, we dissect out part of the tumor, we get patient's reports, and we also get blood to do germline genomic analysis. We can do genomic analysis on the patient-derived organoids. We can grow them and then use them in functional studies such as the drug screen, functional analysis, and cell biology, and we can also do genetic analysis. We're also doing proteomic analysis, but that's not included here. In real life, this is what it looks like. The student will go to theater and stand by while they do a radical mastectomy, which is the standard procedure for removal of breast cancer. And as soon as the surgeon is done from the breast tissue, they'll remove a piece of the tumor. This gets rushed back to lab, processed, and then embedded, because importantly, these are 3D cultures, so we embed them in a matrix. And as you can see in the bottom here, what you get is these cells that grow over time. What's important to understand is that these cells are grown in a media that is defined with a number of factors that are important to keep the stem cells or the cancer stem cells alive and thriving, because that's how we can keep the culture looking like the original tumor. So overall, we've collected 33 samples, of which 10 were successful in establishing these patient-derived organoids, and you'll see we've lost 23%, so we're nowhere near a high success rate yet, and that's also still an active field of investigation to improve on this. But from these, this is just a genomic analysis, so we've analyzed variants in the 50 most commonly mutated cancer genes to show you that when you can see that the patient, so the germline of the patient, the tumor of the patient, and the organoid of the patient nicely clustered together for each patient, showing that the organoid really represents the tumor material that we had. So for all of them, they cluster nicely together, which tells us that we are actually retaining the genetic variety or variation that was found in the tumor in our organoid models. And you can see that here as well with these are some of the somatic mutations you find, and you see that they, between the tumor and the organoid, they have a lot of the variants in common. What's also really important is to understand that when you dissect out tumor tissue from a tumor, you actually have the minority of the cells that you dissect out are actual tumor cells. In fact, when you look in this graph, you'll see on the left-hand side the tumor, and you see here, this is one of the mutations we found for this specific patient, and you see that the NRAS mutation is only in about 9% of the cells, which means that almost 90% or just over 90% of the cells are actually non-tumor. But after having established and grown these PDOs, we now have a pure tumor culture. So all of the cells actually carry this mutation now, which tells us that we have a pure culture. It also tells us that this was one of the driver mutations that is present in all of the subpopulations that are present within this tumor. And so in contrast, you'll see here for these two mutations, they are low and are higher in the patient-derived organoid. This is getting rid of the impurity, so the non-cancer cells. But you can see that they represent two different subpopulations within our complete cultures, showing that we also keep different subpopulations of the tumor cells alive. So not to lose out on gathering data, the patients for who we lost the organoids, we still have the tumor material, so we also did genomic analysis on that versus the germline, and we can do clustering analysis like this and use that for variant calling, which is a sideline, just making sure that we make use as much as possible of the samples that we collect. And then we come to the drug screen. So what we want to do is test the chemotherapies that are commonly used to treat breast cancer and see how our PDOs respond to that. And then at the same time, we'll be following the patients to see how they respond, to see if there's a correlation. And you can already see from the 3 drugs that have been tested here, so 5-FU, paclitaxel, and doxorubicin, that I've divided it up between responders and non-responders at the bottom. And you'll see that there's, for a number of lines that respond as you would predict for the different drugs, but we already have certain lines that are resistant to commonly used drugs. And that's summarized here, where in red are the ones that are resistant to that kind of drug. So even from the small panel of only 5 lines, we already have a divergence in terms of sensitivity towards the commonly used chemotherapies. So this forms the basis to then go look genomically and proteomically to identify markers that can be associated with that we could then use to stratify patients within the public health system to improve their treatment outcomes. One thing we noticed as well when we grew the organoids is that when we let them grow for a long time, you'll see that from the original organoid, you see these tracts of cells emanating. And this has been shown in vivo to actually also happen during metastasis, that you get these tracts of cells moving away from the primary tumor because the leading cells actually remodel the extracellular matrix. Remember, these are embedded in matrix, and they remodel and form these little paths that other cells can follow. This indicated to us that there's some metastatic potential within these lines, and so we wanted to explore that a bit further. And one part that we did here, now I'm going to try. Oh. Let me see if this will work. So here's one line that is a very epithelial tumor line. It's from a patient that had stage 4 cancer, had a tumor that was about 10 centimeters in diameter, but no invasion. And you can see that even if we take these organoids and plate them on collagen, which is done here, they don't attach. Whereas this line, for instance, as soon as you plate them on collagen, they stop being these organoids, get flat onto the cell surface and start migrating. So they clearly have a much higher metastatic potential and are less epithelial. So we further investigated that, and of course metastasis is based on adhesion and migration. And you'll see that when we do an adhesion assay, for some of the lines we get a lot of adhesion after 2 hours here, whereas with the 6767, which you saw in the previous video, that doesn't attach, we hardly have any attachment. And for those that have high attachment, we also check migration, and indeed we see that these cells are very migratory. And you can see that in the confocal as well. When we allow these cells to sit on a flat surface on collagen, some of them will remodel their actin cytoskeleton to undergo basically what is an EMT, whereas some lines do not go through that. So there's clear biological differences in these patients that we can exploit to understand these processes further. So one thing that we've done is to improve our expansion of these lines. We wanted to know if some of these lines that actually by themselves grow in 2D, whether we can expand them in 2D, but then bring them back to 3D, and whether that they then still look like the original tumor and the original PDOs. So first of all, we just expand them in 2D, put them in 3D and see if they grow, and indeed they form organoids like they should. And with confocal, we can also confirm that they normally form proper cell-cell contacts as you would expect in 3D. So they can be an EMT, so be a mesenchymal form, but if we put them in 3D, they'll revert back to the epithelial type. What we show here is, as well, is that, remember I said that the media contains factors that promotes the survival and proliferation of the cancer stem cells. So what we did here is just look at a stem cell marker called CD44, and we can see that a lot of the cells are positive for this marker, which indicates that we are retaining the stem cells in this culture. At the same time, when they are in 2D, they have gone through epithelial to mesenchymal transition, which is distinguished by an expression of this protein called vimentin, which is present in these cells. So overall, we've established PDOs of African breast cancer patients, which is the first, and we've shown that we could use this in functional personalized medicine to personalize the treatment for these patients. We're annotating these PDOs to both broaden our knowledge, but also to really identify these markers for stratification, because this process is pretty expensive. So if we can generate markers that can be used as proxies to determine treatment choices, that will work really well. And then further on, we're characterizing these PDOs to really understand the physiological processes and the difference between what is known, which is mostly based on European patients, to what we have within the African population. Just want to make one last slide here. So these models are lovely, they're brilliant, and they're really interesting to look at, a much better option than cell lines. But we have to acknowledge that there are certain limitations. So they're very expensive. The media is about 2,000 rand for 50 milliliters of media, so it's not cheap. It's time consuming and laborious, and you have to become pretty good at at handling these. The success rate can also be a real problem, especially in the case of breast cancer. The similar thing is for iPSC models. Again, it's a really beautiful model that can be used for a lot of disease modeling, but what a group of us nationally are now busy writing a manuscript for publication in Altex. What we're proposing is that there should be some kind of coordinated model within the restrictive environment that is South African research, where we have national funders as coordinators, a small group of generator labs that will generate these models, and then collaborator labs that will actually make use of these models. That's what we're proposing. Okay, lastly, very importantly, I want to thank the MRC for the SHIP funding for the pharmacogenomics. As you can see, it's a big team that takes part in this because it's multifaceted and we're doing a lot of different things. Importantly, Malosi and Brandon, who are the surgeons who provide us with the samples, Sindisiwe and Pravin are doing proteomics, and Richard is the oncologist, and Theo Heinz is actually Illumina software engineer that helps us with bioinformatics. And then all of this work is really done by Adela Nau, who's an MSc student in my lab, together with Unati, an honors student. And then Khopotso and David are the bioinformaticians that do the genomics. All of this is done within the Center for Neuroendocrinology, and I thank you for your time.
Yes, it's open for questions. Right, we have a question there.
Thank you for a very interesting talk. You had a very nice slide at the beginning of your presentation where you showed the comparison between Black African cancer patients versus the European patients. I'm just wondering, have you perhaps done an analysis between Black African versus white African cancer patients? Do you think there would be differences in terms of the pharmacogenetics that are that vast if there were perhaps African white patients maybe?
Right. No, I don't think so. I think the African white population is quite well represented by European white populations. You have to remember that the white population globally comes from some severe bottlenecks. So in terms of genetic diversity, the African population is 1,000 times more diverse than the white population. So you can pretty well approximate what's done in European or in American white populations to what's present in African white populations. But the opposite is true for Black Africans. So you cannot approximate what is done in African Americans, for instance, with South African Black populations, because African Americans have admixture, and so that makes the whole picture different.
Understandable. And then you had a very interesting slide on, I think it was drug responses as well.
Mm-hmm.
I'm just wondering, since you were doing the analysis on the organoids, how did you actually assess how far the drugs were able to penetrate? Because I assume the core of that cancer organoid would be more necrotic, and then in terms of penetration, did you do any staining to actually show that the drug actually got to the core?
That's a great question. So the way we do this is we actually establish single cells from the organoids, allow them to grow to only about 8 to 16 cell stage. So they're very small when we do the drug screens to prevent exactly this problem of penetration. Of course, that's not entirely what happens in vivo, but we can't, if we'd have to, the models don't exist yet to really recapitulate the vasculature in the tumors, which would be really important to be able to really assess that. So the intermediate is to use small organoids so that penetration is not a problem.
All right, and then just a final question. So the organoids in the plate itself, I assume, like, how do you ensure then that once they're exposed to the media, like the bottom part and the top part is, like, it's fully exposed to the drug? Because I assume the top part and the bottom part will have different types of exposure, right?
No, so it's done in a 96 well, and so the drug is everywhere. There's complete exposure, so it's homogeneous throughout the whole media. So there shouldn't be any gradient effects, if that's what you mean. There won't be any gradient effects.
Okay, thank you for that interesting talk. It was quite scary to hear that that this black or African woman actually experienced this breast cancer at premenopausal stage, and the survival is that low. Now, rising from the people from which the samples were taken, I'm just wondering, what is the team doing in terms of community engagement? Is there anything in terms of raising awareness or preventive measures that we have embarked in terms of giving feedback to the community that gave you the samples?
Yeah, that's a really important question, actually. And in this project specifically, we don't really have a large community arm to this project because it's so theoretical and quite difficult to explain. We do, however, have another project where we're looking to establish liquid biopsy technology to actually be able to do early detection of cancer at district hospital level rather than tertiary hospital level. So there we do have that, so that we can explain to people that we can actually improve their outcomes by detecting cancer earlier. It's a very important question, though.
Hi, thank you. If I understood your question, your talk properly, your organoids are more successful in the non-metastasizing cancers, right? The non-migratory, the migratory, non-migratory cells.
Yeah, so I'd have to say we don't have enough samples yet to really be able to say that, but I would say overall, the ones that are more aggressive tend to grow better, yeah.
And then, can I ask you, so I understand that the center of any organoid is a little bit necrotic. So for me, that means that it could influence the signaling you get from there. So, and this may be a stupid question, but is it possible to potentially use a scaffold around which to start constructing your organoid to kind of not have that, or do you want that? Is there a reason for having this necrotic core?
Yeah, that's always a discussion people have about this. So I think we have to be clear that if, like, when we go to theater and we open up those tumors, most of them have a huge necrotic core. So I think having a necrotic core in the organoids is maybe not that bad because it does actually represent the in vivo situation. Nonetheless, for some of these analyses, it probably is better not to have it. And so at the moment, the way we deal with that is by not growing them very big when we do the assays. When we grow them to expand them, we don't mind a bit of necrosis because you get rid of it as you passage them. But for the analysis, we try to avoid that. Especially for things like proteomics, we definitely try to avoid that. Yeah.
I just also have a question. I just want to know your organoids, when you extract it, how does it evolve over time? Do we keep its genetic phenotype, or
yeah,
mutates?
All right. So the data that I showed on the purity and then the subpopulation. So between the tumor and PDO, we only analyze the PDOs after at least 3 months of growth. And there's a very important reason for that. Those of people that have worked on primary cells will know that you can take cells out of an animal or a human, grow them for about 10 to 20 cycles, and then they die. And we see the same thing here. So 80% of the sample is not tumor, so we need to wait to select out and kill those cells, and then we want to know whether they're stable, so we keep them growing for a long time. So we go through a crisis phase as well with the PDOs and have to expand them from that. So when we do the genetic analysis, at that point we can say that these are pretty stable features. Yeah, we do lose some features, we can see it in the genetic analysis, so some subpopulations are lost, but overall I would say the majority of them are kept intact. Thank you very much.
Thank you, Prof. Okay. Dr. Sokota. Dr. Sokota will just give us the logistics following on into tea time.
Thank you very much. Please give yourself a very round of applause. You've been a very good participant. Okay, the logistics are as follows. We're going to do the breakup sessions. This is what we call the warriors, and the next one is what we call the daily lily. So we're going to have a cup of tea, and then at 20 past 10, we resume our session. And ladies and gentlemen, we are trying to keep to time as much as possible because our program is fully packed. Please enjoy the tea, and then when the bell rings, it means it's time to resume our presentation. Thank you. Professor Ntsipa, please approach the desk. Professor Ntsipa.